the fifth episode of the HMAI speaker series. Today we have Adib Fallahpour. Adib is a research lead at the ARC Institute. He's also a researcher at the Vector Institute at the Wang Lab, where I was two summers ago. He is also an undergraduate student at U of T, studying computer science and neuroscience. Welcome, Adib.
Yeah, thank you for having me. I'm going to talk about BioReason today, which is our NERV's work on creating a multimodal reasoning model in biology. And it's been a great work of a lot of people from like both U of T and Vector and ARC and UHN and like a bunch of different places. And I'm very excited to kind of walk you over how we thought about this problem, what we did, and what is exciting about this work. So essentially, the goal here is to develop a multimodal model that can reason about mutations. So if you have a DNA input that has mutated, you can understand the effect of that mutation, but also you understand the biology surrounding that mutation, like understanding the genetic pathway, the proteins that were mutated, and the connection between these two. And ultimately, can you evaluate this against the LLM only or DNA only approaches and see what works best? Just for some background, we're all familiar with large language models, but I want to give you a sort of a perspective on ultimately the fact that these LLMs are giant embedding models. Like the only thing we're doing is that we start with some interesting generic input embeddings that are maybe token level generic embeddings. We maybe add some positional embeddings and a bunch of things, but ultimately what we're getting out of this is again a bunch of token embeddings. Now, whether we take the last embedding and we use that to predict the next token is the only a very small fraction of all the computation that the forward pass does. It. And so this kind of shows us that the embeddings are key. It's about this understanding that whatever this model is doing is just learning some generic embeddings first, and then also learning how it can contextualize those embeddings through layers of computation such that it will make it easier to understand and predict the next word. So if we could take in a DNA model instead, we also have a similar approach happening where we train DNA foundation models or protein foundation models with the same approach that take DNA sequences, consider them as a bunch of characters, and then do autoregressive training on to learn proper embeddings for the DNA sequence. So this will give you kind of some insight that understanding DNA is really about understanding good DNA embeddings, which happens in the DNA foundation model due to just the enormous amount of data that these models process regarding DNA. And understanding language happens to LLMs because they do process lots of language data. But if both of these are giant embedding machines, then there should be a way for us to just swap embeddings from DNA models into LLMs and be able to transfer the knowledge that the DNA foundation model has somewhat learned to a language model that only knows the biology behind it. And the third piece is reasoning, where we essentially consider a reasoning large language model that can think about a question, and we use reinforcement learning to just see which samples will do better and do more of those samples in the future. So if you put all these things together, that's where we kind of get the Bayesian architecture that I'll walk you over. So the idea here is that we have a genetic pathway. For example, you can see this network pathway, and we have all the genes in it. And one gene is here is mutated. We have the exact wild type sequence and the mutated sequence of that gene. And the idea here is that can we predict what is the biological effect of this mutation? Does it lead to a disease? If so, how does it lead to a certain disease? And what is that disease looking like? What we do is we first take the DNA sequences through EVO2, which is our DNA foundation model, totally frozen here, and it will embed each of these nucleotides into an embedding vector. So if you have 2,000 like nucleotides in that gene, you end up getting 2,000 embedding vectors. We get that for both the wild type gene and the mutated version of it. At the same time, the question itself will get like tokenized and embedded, a bunch of embedding vectors per each token. And so you'll have a bunch of DNA embeddings and a bunch of language embeddings, and you just stack them together into this giant embedding out input. So here the blue ones are DNA embeddings and like the purple ones are language embeddings. For the model, it really doesn't matter what these are. The only thing that matters for it are that these are nice embeddings, and it will try to leverage these embeddings to be able to predict and like do well on an autoregressive task. Here, the task is create a reasoning trace that will think about this mutation, going through how this mutation changes the gene, changes the protein, how does that change in the protein might cause downstream changes in the genetic pathway, all the way to predicting the downstream disease that will lead to here being Parkinson's. And this will lend itself to doing a supervised fine-tuning approach of like teaching the model to generate these reasoning traces. And then reinforcement learning, where we use the disease as the reward signal to be able to find and upsample the generations that more correctly lead to the disease that we're interested in. So for some of you who like more math, we can think about this as we have a DNA foundation model who just takes in genomic sequences and it will embed those to a bunch of embedding vectors. And we have a language model whose goal is to get a textual query and just maximize the essentially the log likelihood over the answer it has to predict. And the DNA-LLM integration then becomes an input of DNA and text embeddings together, where we'll stack these embeddings, doesn't matter how many we have, and we'll put this in the context of the LLM, and the LLM will process that for our target training objective. For the reinforcement learning side, we'll just do the normal GRPO approach. Here we can specifically use Dr. GRPO, but essentially it's the original, it's very similar to the original GRPO approach that we don't have any value function. We just have the reward signal that we use. And here our reward functions is mainly the correctness reward, which is about did you get the disease right or not? Conciseness reward, just to make sure that the final answer is short. And format reward, which is about just ensuring you use the thinking blocks correctly. So very simplistic reward approach that will only try to maximize what generations will lead to correct disease prediction. For our data sets, we ended up using data that actual biologists had prepared. So there's a variant network database called KIG, where you have essentially all these genetic pathways, their gene names, and specific ground truth of the biologists, like kind of like sitting and writing down exactly how they think a certain mutation in this genetic pathway causes a disease downstream. And so we take this ground truth of data and we just put it into a question answer format, where the question is about here's the sequences, here's some information about the pathway, and the reasoning trace is just a step-by-step version of the ground truth biologists had prepared, where we kind of break it down into analyzing this ground truth in reasoning causal steps. And ultimately, the model will predict the final answer that will talk about how it thinks it will lead to that disease. This will give us about 1,500 QA pairs from 300 networks, which covers 37 different diseases. So it's not that much of a data, but it will help the model get started. On the other hand, we also have some variant effect data, like usual, where we have no coding variants and coding non-SNV variants. These are all just single nucleotide or single nucleotide or non-single nucleotide variants, where essentially there has been some mutation, and the goal here is to predict whether that mutation is pathogenic or benign. So on the KEGG data set, we see quite remarkable performance when you combine DNA and LLM together. So if you start with like nucleotide transformer, which is a DNA foundation model, or EVO2, which is a more powerful DNA foundation model, and put a classifier head on top of their embeddings. So freeze the DNA model and just use an MLP head to predict the disease based on those embeddings, you get a pretty decent performance. These models will kind of like learn the different genes and the different pathways that they might be involved in, and kind of use those DNA embeddings to be able to accurately predict the disease. So you get like 86, 88%. If you bring a Quen model as a language model that takes in the DNA sequence directly as raw text, it will still kind of do good as well. And here's essentially feeding on its understanding of the genetic pathway. It might see like different gene names that it might recognize, and based on those, it will reason about what is the disease going to be, because it doesn't have much of an understanding of exactly how that mutation changes, especially if it's like a codon changing to something else. So here you get like 85, 90% performance. But then when you combine these two, for example, you can see when you have Quen and Evo2 together, you get like 95% compared to Evo2 alone, which was 88, and Quen alone, which was 90. So you're boosting performance on top of both of these, where this model is now able to leverage both this understanding of DNA and this understanding of language to do great on this downstream task. And if you further on use GRPO to do some reinforcement learning, you can even get a boost up to like 98%, essentially solving this benchmark. The other things you can see is that kind of scales with model size. So like Quen3 1B does slightly worse than Quen3 4B on this task. And at the same time, like the Evo2 variants with Quen3 1B do a bit better than the nucleotide transformer model with Quen3 1B. So this shows that the better the DNA model, the better the language model, and the better the sort of a joint learning of these two happening together, the better performance goes. And we can still boost that further with reinforcement learning, but the gains of reinforcement learning are higher on like the 4B model compared to the smaller 1B model. On the other hand, you can see a case study where we have like a genetic pathway about the PFN1 gene on this pathway. And the question here is that, oh, we have a mutation, the DNA sequence is not shown, but this will be passed to the model. And the idea is, can you predict what is the disease and how does it happen? And so on the KIG ground truth, you can see like we talk about what is that variant, how does it change the gene, how does that nucleotide change causes a downstream change that causes like ALS, and that's our final disease prediction. And we can also see BioReason's output, which is a slightly different way of reasoning about the same disease, where the model has predicted that this mutation will cause a change in the protein structure and function of profilin 1. So it understands what that gene is. And it thinks about the role of that gene based on its function in regulating actin dynamics. And based on that, it will decide if mess up the actin dynamics, then the muscle movements might be impaired, which could lead to ALS. So it's a slightly different way to reason about the same disease, but it's again correct. It's very interesting to see happen. On the other hand, we have variant effect prediction, where the main difference with the kick side is that there's not much context for the language model. So we essentially have only input DNA, and we're asking, is this mutation pathogenic or not? There's no sort of context about the genetic pathways involved, etc. And the idea here is that can the model still do better with the DNA embeddings as a language model than like a classifier head? So you can see an LLM only model will barely do random performance. And that's because it's very hard to look at a mutation that's like in the coding, like a codon is changing and be able to reason what is that mutation doing, whether it's pathogenic or benign, if you really don't understand DNA. A DNA model with a classifier head on top of it, like a binary classifier head, can do relatively well, I guess like 60 or 70%. And that's pretty much what we do when we're training DNA models. Like we train DNA models, we freeze them, and we might train a classifier head and fine tune that for like some downstream variant prediction tasks. But when you combine the DNA-LLM embeddings, you suddenly get huge boosts, even up to like 80%, where now the LLM can leverage Evo2's embeddings much better than a classifier head and actually deliver a much more accurate prediction of whether that's a pathogenic or benign mutation. You can see the same thing for like non-single nucleotide variants. Now, when it's non-single nucleotide, sometimes the LLMs can kind of guess by saying there's deletions or there might be like a huge chunk of the DNA changed, and based on that, try to reason how things work. And that's why we can see like a relatively good performance. At the same time, DNA models can also like look at how the DNA embeddings have changed and be able to reason as using the MLP classifier head they have. But when you combine these two, you get like 88, 89% performance with their combined model. And this kind of shows that the ability of the language model doing better on DNA embeddings will come both from its understanding of language, but also from just it being a much more powerful model in making DNA embeddings processed further such that they could be ready for doing variant effect prediction. This kind of shows us that the DNA embeddings we are getting are not very suitable for variant effect prediction directly, and we need models that can actually unravel them into linearly separable embedding spaces for these tasks. And that's why like the benefit of BioReason kind of feeds on both of these, the ability to leverage DNA embeddings better and the ability to bring LLM's understanding of biology to techniques. So overall, the sort of contributions have been, we created a sort of novel multimodal architecture as the biggest integration of a DNA model and an LLM. We also showed that you could use multimodal reasoning on like creating a model that can actually develop new reasoning interpretations. We developed a reasoning benchmark called KEGG, and which kind of solved. We also showed that empirically you get up to like 15% performance boost over like a span that's like for variant prediction is essentially a 50% span, which is pretty huge. And doing all of these is also giving you a reasoning trace that you can actually look at and kind of understand what the model is thinking. Very small and preliminary work, like the scale of data is super small, but it kind of shows that the core hypothesis that we could create multimodal models in biology and we could have them do reasoning actually works. And it will give you a substantial boost over the LLM only or DNA only approaches. Some of the learnings I'll summarize is that surprisingly, DNA model embeddings work better when they're not from the last layer, but we're like kind of like 70, 80% through the network. I think it again kind of boils down to the fact that very last layers of a DNA model are being kind of changed to be linearly separable for predicting the next DNA token. But our goal here is to understand a representation of the DNA. And so layers a bit earlier in the network work better than the ones who super at the end. LLMs are very bad at raw DNA processing. If you just bring in ATCG as like text, they're not really good at even understanding like codon changes. And just because of not having seen a lot of DNA during their learning, and also the fact that tokenization, like the BPE tokenization we use for LLMs, will just going to totally ruin the DNA tokenization that should really be character level to understand things. Reinforcement learning needed lots of compute. Like from BioReason 1, we've done a lot of work to make it super efficient. And Evo 2 is a better model in DNA understanding than Nuclear Transformer. Of course, we need more data and harder data. And of course, DNA LLM learns surprisingly well. And I'm still open to intuitions about why it learns better. Like there are intuitions about like, yeah, he understands language, or the fact that it's a much better classifier head sort of model as a language model. But I still think there's a lot of unknown things here. Essentially, the main thing is both of these models, like in all the baselines, the DNA embeddings are frozen. Like we're not having the DNA encoder learn any pattern, any new patterns. Of course, if you allow the DNA model to learn new patterns, things change. But with the same representations, the LLM is able to do surprisingly well. And I'm not really sure how that happens. Like on one hand, we could argue that it kind of means that the representations these DNA models learn are quite fruitful, and they're actually in like they actually involve lots of information that we just need to extract. But on the other hand, it also can show us that maybe LLMs are just naturally great at even further processing that DNA sequence, even though the DNA encoder is frozen. Our vision essentially for BioReason series to create AI system that can reason in biology from first principles. Here we worked on how mutations lead to disease. In BioReason 2, we actually are working on how proteins function. And in BioReason 3, the goal is how drugs affect cell state, or essentially how does a virtual cell model can reason. So our next steps is to release BioReason Pro, our next model in the series, probably in December, where we kind of showed that a model that can take in protein embeddings, some context about a protein, and as a language model, can outperform other models in protein function prediction with a step function improvement. In BioReason Cell, our goal is to create a virtual cell reasoning model that can actually leverage both the language model understanding, a cell model's understanding, maybe even DNA and protein embeddings to be able to reason how perturbations cause changes in the cell. And we've had very interesting partnerships between like ARC, UHN, Vector, and other places. Our team is kind of scattered across both Toronto and San Francisco, and we're all working hard to create the sort of next frontiers in the bioreasoning sphere. And I think the main aspiration for this project has been like one of Jensen's quote about how digital biology is going to be the biggest opportunity in the next decades. Essentially, the idea that biology could finally have the opportunity to be engineering and not science. A lot of the things we do in biology are just science. We do drug discovery. We throw darts and hoping some drug lands or some target is found or some clinical trial passes. But in reality, we should really do engineering. We should do drug design. We should actually go and understand the systems fundamentally to find the right targets, the right drugs, and the right clinical trials for them. And I think this is going to be a very huge opportunity for models like BioReason or paving the way and further models to come to allow us to truly leverage all the data we have about biology and create cohesive narratives, allowing these ML models to finally understand the biology and be able to create causal links across it that even though we have the same data, we don't really understand yet. And here's like a project page, our GitHub, our data, and anything else that you're interested in. Ping me up if you have any sort of questions or opportunities to do things together. And thank you for listening. This is QR code for my Twitter in case you want to follow.
Thank you so much, Adib, for the amazing talk. We have time for some questions, but I can go. I can go first. Can you go back to the slide where you were showing the architecture, like the second or third slide?
This?
So, yeah. So here Quinn is not frozen, right? It's
No. Quinn is actually getting like Quinn is the main thing that gets trained. It's getting trained using LoRa, so it's a really lightweight LoRa. I think that was rank 16 or 32. And the other thing is getting trained that I forgot to mention is also the projection layer we have. Like we need some sort of linear projection from the embedding dimension of Evo to the embedding dimension of Quen just to make sure they kind of fit in. That also gets trained alongside the Quen model.
so how compute heavy is it? Because there's a lot of tokens that come from the DNA model, right?
Yeah. So this is actually not super compute heavy. Like I think I used 1H100 for
Mm.
any of the experiments. Like all the experiments kind of fitted on 1H100. The things that would make it compute heavy is if you want to go fit with a bigger language model size, of course, you will need to fit things in the GPU. And I think beyond 8B, it will be very challenging to fit into a single GPU. The second thing is if you have like very long DNA sequences, then you get lots of embeddings to put into the context. So like empirically, when you want to do like an 8B model training with a good batch size, you could actually fit up to like 10K tokens in the context. So considering that like your question answer pairs will probably be like most of it, it will leave only a few thousand for the DNA input. It will cover most DNA sequences, like having like 2048 nucleotides, like about 600, 700 amino acids is kind of good enough. But if the DNA sequence is longer than that, then you either have to truncate EVO or just have use more compute to be able to fit in all those. The good thing about nucleotide transformer is that it has like a 6-mer tokenization. So it will be essentially 6 times more efficient in representing in the number of tokens it needs to represent the data. So with like nucleotide transformer, you can actually represent like a DNA sequence of 10K nucleotides if you want to with the same framework. But when you use Evo, because Evo is character level tokenization, you get lots of vectors. So there you have to either downsample in a way or just like increase your compute to be able to fit it in.
Okay, so I think this architecture is nice because um, it doesn't add any additional like actual model like a decoder or anything. But have you guys explored other ways of combining the text with a DNA embedding, so like a cross attention thing that you can add in the LLM layers, or any other way to integrate the tools that maybe
Yeah,
swap?
that's a good question. I think we were mainly inspired by how vision language models work. Like the same problem we have here is what people had with vision, that you have a vision encoder, maybe like a VIT that can process images, and somehow the language model should take in its representations to be able to understand an image and like caption it. And I think the way people in vision, so like very original vision models were yeah, like kind of trying to do some cross attention or some like funky stuff. But then later on, we realized they could just pass in the embeddings of the image directly in the context of the LLM, and the LLM's attention layers will learn to deal with that. And I think that's why we kind of ended up doing this as well, because this is a much more simpler and elegant approach. It doesn't add any further like cross-attention complexity. It will just essentially be like swap the DNA embeddings from like the language model with the ones from Evo. Uh, I do think there are a few ideas quite unexplored here. Like one is the power of contrastive learning. Like in the LLM, like in the VLM space, we do a lot of like clip style contrastive learning, maybe here and there to align the space of the vision with the images, with the text. Here, we don't really do something like that. And the main reason we didn't do that was because, well, on the one hand, we want to keep it simple, but on the other hand, it's very hard to like properly do contrastive learning here because of the data. Like in images, you have lots of captions and lots of images. And a lot of the time, captions and images are very, like, they're sides of the same coin. Like, they're kind of representing the same thing in two different modalities. So clip style training there makes a lot of sense because you want to like unify these. But a DNA is quite different from language. There's no sort of caption for a DNA that you can use for contrastive learning. You could do like contrastive learning of trying to push all the DNA sequences away from each other, like stuff like that. But it just doesn't really feel right to use eclipse style training for aligning two very different modalities that you really can't have like matching pairs to use for clip. That's why we didn't do any sort of contrastive learning, mainly focused on just do just put embeddings in the context. I think the ultimate form of this is when your language model has just natively been trained on DNA so that it can actually process DNA tokens as native tokens. That will also allow you to generate DNA because now the DNA tokens are available in its vocabulary. But that will be like a much more compute intensive approach because now you have to like train on lots of DNA sequences to actually enable your language model to understand them. And you might even start losing some language understanding when you do that.
Okay, makes sense. So this is basically lava then, but instead of images, is DNA. It's
Yeah,
exactly the same.
exactly. Even simpler than lava. Like
Yeah.
this is very minimal.
Last question. There's some questions in the chat, but last question for me is go to the second results you're showing for the mutation. Second table.
This one?
Yeah. So the model sizes for the DNA only models are quite small compared to when you combine both, right?
Yeah.
So is there larger models that you can try, like same scale as the NT plus Quinn? Actually, that's not that much larger. Never mind, because that's like a 1.5 billion only. Uh, but then
Yeah.
the last two rows are like that would be 5 billion and 2 billion.
Mhm.
Uh,
Yeah.
so
That's a good point.
yeah,
So I think the main thing here was that we wanted to focus on seeing the trend. But I definitely agree that like we could have done Equine 3 8B training. For buyers until we have like 8B training. Um, on the DNA side, it gets a bit more challenging because like the next available size of Evo is 7B, and 7B is like big because like Evo is you could have maybe pre-compute like the embeddings and you don't need to run Evo during training because the embeddings are frozen, but then during inference, you really will create this sort of gap that you have to first run Evo, then cache the embeddings, then run Quen to do the generation. my intuition tells me that, of course, if you go from like Evo to 1B to 7B, we should get better. And we definitely see that Evo to 7B is much better at variant prediction than 1B. But on the other hand, like if you make the Quen model bigger, that means like more biological understanding of Quen, and so just essentially just better training. But I'm not really sure if we train like an Evo to 7B with Quen through 8B here, we'll see much better performance than like 80 or something percent. And that's because variant prediction is like at some point you kind of saturate the performance. Like the rest is kind of noise. It's not really straightforward to predict them. And and then I think it's not really a matter of model size here. It's more about a matter of data. That's like, is your training data diverse enough to teach the model all the different things it can see? Because the DNA models has already learned so much DNA, they just needs a proper alignment to kind of like learn that separability for the downstream task. But yeah, it's I think it's interesting to kind of evaluate the scalability, like draw some scaling curves for the encoder and the language model and see like how do they differ and what sort of combinations do you get the best output.
I mean, not just that, but I'd be curious to see if you hold constant the number of parameters in the DNA only model and the DNA plus LLM one. In your DNA only model, you have Evo, it's 1 billion parameters, right? And then the DNA LLM
Yeah.
model, it's double that with Quinn. So
But
has
the
more.
thing is the LLM is mainly frozen, right? Like only the LoRa parameters are getting trained, and there are not that many parameters. So essentially, the difference of the number of training parameters between two setups is quite small because the LLM on the DNA only will have a classifier head, it's not just a linear layer, it's like an actual MLP. And the Quinn model will have, essentially this LoRa. The LoRa, I think, is like rank 16 or 32 or something. So it's not that many parameters when you consider that. So when you compare these two, you actually have a very comparable amount of trainable parameters. Um, but yeah, like in terms of total parameters, there's a big difference.
Yeah, that makes sense. I like some questions in the chat.
Oh, let me see the chat question. Have you tried not frozen evo for example, also at LoRa? That's a good point. We did not do a proper experiment where we kind of like unfroze Evo and see what happens. But in some low-key experiments we did with Nuclear Transformer, we actually saw that performance goes up when you do train the DNA encoder. And I think it's because the model is able to like learn new patterns for like the DNA understanding that will help it for this task. On the other hand, it could also be that like very early on we get better representations, then we even give more room for the language model to do better. But we didn't fully explore it because most of the time when you're dealing with these systems, you actually freeze the DNA encoder because you assume that the DNA model has so much DNA that we don't want to like make it overfit to a specific task. But there are cases, in my opinion, that will definitely make sense to do fine-tuning of the encoder. Like for example, if you're training, like for example, for bars and cell, where you're kind of training a cell foundation model jointly with the language model, it would make a lot of sense for the encoder to also get revised a bit. Um, Not measurable. That's a good point too. I think the main reason we didn't merge is because we want to evaluate different, like essentially we want to focus on different problem statements. When you like for example, protein function prediction, it really is not relevant to DNA understanding and effect understanding the effect of DNA mutations could be better done with proteins, but it's also kind of implicitly learned mostly from just the effect on the pathway rather than the effect on the protein itself. Because in most of the scenarios like this, you're like, oh, the protein is going to change, and we just want to reason how does that change cause the change in the pathway. Our vision is to truly bring all these things together. Like you could have a single model that can take in both DNA and protein and like cell embeddings at the same time, and that's just a matter of how good a data can teach them. But the main challenge there is that if you want to jointly train all of these together, you would need data that would allow that. And there's a very small amount of language data that can also span multiple modalities. Most of the time, just have language with just one of these modalities. And so that's why we kind of did like separate bars and trainings for different hypotheses. But if you want to bring them all together, it's kind of possible now that the enterprise set up. Um, I guess it kind of depends on what's the task that you have in hand that will really leverage all these embeddings. But yeah, it's not really far off. Then in the future we'll have models that are omni-modal than just multimodal. Yeah.
Okay, any other questions? Okay, if not, I guess we can end it here. Thank you so much, Adib, for joining us today.